And our next speaker is Collin Ewald from ETH Zurich.
Great, thank you so much. I'm happy to be here, and I want to actually follow up Adam and Taby's talk and the question that was raised by Jan Wick about the senescent cells in C. elegans. So we have been working on that for the last 5 years, and we got some answers there. And so you're perfectly right. I mean, C. elegans is a post-mitotic organism, and we were asking whether, they has senescent cells, and we used the beta-gal staining, and what we see actually, we see a really nice increase in beta-gal staining during aging, but not all longevity intervention actually slow that accumulation. And what we found is actually that metformin works to suppress the beta-gal staining during aging, and that works via actually RAC ATPases and nucleocor complexes is an Asian pathway that signals to the mTOR signaling pathway, which was very interested. And then we thought, okay, can we use this as an in vivo screening system? Because, it's C. elegans. I mean, it's not the same senescent cells. And so we done a whole screen, and we actually found reganase, which is an important regulator of ETS4, transcription factor, that completely suppressed that. And then we showed that in collaboration with the Marius lab at the RIBA, that it actually works also in human cell culture, right? So we established that system. The paper just came online yesterday in Shearer Science. So please look it up. The first author is Richard Wentz in Shearer Science, and I'm happy to take any questions about that senescence and C. elegans. So with that, I'm going to shift over to the extracellular matrix, and the question we're asking here is, do longevity interventions actually repair age-related collagen cross-linking? So I take a little bit back to give you a little bit background. Oops. Sorry. So, I'm sorry. Did I do something wrong? Okay, so we look at conserved interventions that slow aging. We know that reducing insulin one signaling, caloric restriction, and mTOR signaling actually slows aging. And we know that certain processes inside of the cells are repaired, like DNA repair, epigenetic regulations, and also proteins inside the cells, either in the mitochondria or in the unfolded in the ER, or then in the cytoplasm. However, we lack a complete understanding how proteins outside of the cells are repaired and maintained. And the major proteins outside of the cell are collagens. Collagens are secreted and form these net-like structures that surround the cells. Now, it has been said that collagen is the most abundant protein in your body, around 30%. And so that should be the same for mice. And when we looked at it, we couldn't find any original references who had actually determined the collagen content of the whole animal. So what we done, we took whole mice, put them into a blender, lysed them to the amino acid level, and actually compared how much total collagen over total protein are in mice. So if you're female mice, it's 12% of your total protein. If you're male mice, it's 17%. It's not quite 30. And the interesting part is a gender difference, which we don't understand why, a female mouse has less collagen than a male mouse. However, it's still a very big abundance, right? Meaning that how are these proteins then repaired and maintained during aging? And so just to give you background, what is an extracellular matrix? Extracellular matrix come in two kind of flavors. One is a basement membrane sheet, which is shown over here with shown here. This is the sheet-like structure, and on top of it, for example, is epithelium cell. And the second structure are these connective tissue, these collagen fibers here. Now, you're very well aware that inside the cells it's very crowded with lots of proteins, but the same is actually also outside of your cells. So what you see here is the cell membrane, and then you have collagen receptors like integrin that bind different collagens. So here would be the basement membrane, and this huge bundle here is a collagen fiber. And so you can also imagine if you're a molecular growth factor, how do you navigate through the external matrix? And so there's lots of information going back and forth and has a lot of different functions. Now, why do we care about the external matrix? It all started in C. elegans when I did my postdoc, and that was also collaboration with Colleen Murphy's lab, where we looked at actually long-lived C. elegans. So in plaque is the wild-type lifespans. You can double the lifespan by just reducing insulin IGF-1 signaling. And if you knock down some key collagens, it actually almost completely suppresses their longevity. Furthermore, when you overexpress the same set of collagen, it actually was sufficient to increase the lifespan of C. elegans. So if something is required and sufficient, that means there must be a mechanism behind it. And in my lab, we try to hunt down that mechanism, and we call it external matrix homeostasis. So again, the collagens are outside of the cell. Now, how do you repair these collagen? It's like a fish network. So if something is in there, you need to go and cut it out. So these are the metalloproteinases. They cut out, they degrade it, but then new collagen has to be synthesized, right? So the gene synthesizes. And so this is a process. It's a back and forth outside, inside, and inside-out signaling to really maintain this. And we call this external matrix homeostasis. And as many processes during aging, this external matrix homeostasis is lost or declines. And longevity interventions somehow just keep it longer on or reinitiate this process. Now, why is this important? Because if collagens are not turned over, it's a pretty dynamic system. If they're not turned over, they become fragmented with aging. So metalloproteinases, if they just go out there, they just start cleaving the collagens, so making the matrix mechanically weaker. At the same time, what's happening, there are sugar adducts that bind to lysine and arginines in the collagens, and they're called the advanced glycation end products, and these start to cross-link. So it makes a more stiffer external matrix. So basically, with aging, you get the mechanically weaker, but at the same time, a stiffer external matrix, and that changes signaling, and it changes mechanotransduction, how the changes, environment is read out by the cell. And the last thing that's happening, that's also protein that start to aggregate in the external matrix, the waste products. So now, why is that important? I mean, there are a lot of different things that if your collagens are not properly synthesized, there are a lot of human diseases associated in phenotypes. I just want to give you one example of collagen 2A1. Mutation that collagens are associated with over 250 human phenotypes, right? So just one collagen genes can have a vast majority of different phenotypes. Now that's also the same for diseases. Some of these mutations or SNPs are associated with human diseases, and we identified 333 external matrix genes that are associated with human diseases. And you would assume most of the diseases would be connective tissue diseases, right, or disease, you think about it. To our surprise, actually, a majority of the diseases were age-related diseases, and these are these light green dots over here. Now, why are these age-related diseases so much affected? And what we think is going on that when you're young, you can deal with a faulty external matrix because of the better protein homeostasis. However, when you're old, things break down, and then really this disease phenotypes start to progress. Now, showing you this means also there's a huge medical need, because at the moment there are 8 targets out of the 333 external matrix genes and 27 clinical trials going on. Most of them are actually in fibrosis and in cancer, right? So there's a vast maturity we can study and move forward. Now, to make any insights there, we took a playbook page from the technology tree. So that was something started by the Foresight Institute and Aaron King. I think he's here, is also came up, with this idea. And the idea of a technology tree is to build out a strategic plan, what's going to happen. So in a massive paper, so this is a 20-page paper, we describe what are all the diagnostic tools. You can see the external matrix or changes in the external matrix, then how they're linked to different external matrix components like collagens. Now, what's happening during aging, like the advanced glycation end products and inflammation and everything like that, and then the diseases. And then also try to map out also the different intervention that's possible. So we just mapped the landscape. So that was the first step there. The second step, we wanted to figure out what is the human molecular disease network that governs the extracellular matrix in aging? And we mapped it and linked these different extracellular matrix to intracellular signaling pathways, and it goes down to transcription factor. We are very well known in the longevity field, like, NRF2 and FOXO, right? And that led to the ideas like, what is actually a youthful and healthy extracellular matrix? And I'm not going to show you this. I'm going to skip that part. But basically what we found, this ECM homeostasis part is regulated via mechanotransduction. So physical forces that are transduced in gene expression, and we model the gene expression there. The way we looked at this was very in a systems biological approach. So Alexander Nappa has defined the human matrisome, which are all the extracellular matrix genes out there, and we did the same thing for C. elegans. And basically, it was very interesting to see, because every cell, and that was Tony Viscori's talk yesterday, right? Every tissue has a different expression or proteins that are out there, right? We see the same thing with the extracellular matrix. And we took it actually a step further, because in the field there was a saying that, every cell makes its own extracellular matrix. And so we used single-cell sequencing and looked at this in chick limb development and also mouse limb development. And we were able to use the extracellular matrix genes, around 200 extracellular matrix genes that form the extracellular matrix, which is 2% of the whole transcriptome in single-cell sequencing. And we were able to predict cell type, right? I'm just going to repeat this, only with 2%, right? So there must be some biology behind it. And so basically, the extracellular matrix, you can not only determine cell type, but you can also say what differential state they are and what fate they are. And we know from cancer that if you just look at the extracellular matrix composition, you can predict cancer type, right, and what stage they're at. And so that led to the idea of the mature type. The mature type is a snapshot of the ECM composition, right? So different ECM genes that is associated or caused by a phenotype or physiological state, right? So physiological state could be health, disease, aging, longevity, which is a really nice signature, because it led us to think about, okay, can we take the human mature type, right? So we looked at GTEx data that was, during aging from the age of 20 to 80, different tissues, and we looked just at ECM composition. And we defined a young and an old one. And then we used the young extracellular matrix gene expression to do actually drug prediction. And so with that, we came up with 180 geoprotective or longevity drug, just based on the extracellular matrix composition. And then we tested these in C. elegans, and basically we figured out, okay, this drug extended the lifespan, which was interesting. Now from that data set, okay, we have these predicted drugs. I just want to show you one follow-up, because we took this even further with Meibel biochemistry in a pilot clinical trial. So we gave 32 subjects that had sun-damaged skin this compound Tragobel, which is an extracellular matrix activator, 2 milligrams per day for 2 months, and we saw improvements in skin elasticity, moisturization, and also still skin composition, right? So this is very interesting. So you can use extracellular matrix composition to do drug predictions. Now, this is cool for drug discovery, but the original question I posed here, so this was more the introduction, do longevity interventions actually prevent or slow or even reverse collagen cross-linking? And the way we look at this is we take mice, so either 3 months or 24 months old mice, we take their tail and take the tail tendon out, which is basically all collagens. And it's a very old assay from the 60s where you put the weight onto that tendon and put in urea. So that thing will the collagen will denature and break. And the more cross-linked the collagen is, the longer it takes to break right? And we can measure that. So if you look at 3, 6, 12, and 24-year-old mice, you see an exponential, this is log 2, an exponential increase in the time it takes to break the collagen. So you're assuming that they're more cross-linked. And there's a sex difference in there. Now, we were asking if that is almost, exponential or log 2 linear increase. When you plot it here, this is time it takes to break these collagens, and this is the age of the mice. If we do now an intervention at 18 months, so the mice are pretty aged, they accumulate collagen cross-linking, you can see this. What happens if we do now a longevity interventions, right? And there's at least three possibilities. Either it just slows the accumulation of the cross-linking, or it could halt it, or even that would be best case scenario, reverse it, right? That would be extremely cool. You have some repair mechanism going on that we remodel the extra matrix in the collagen and repair it. Now the way that we done this, we gave, and that was a collaboration with Marcus Rick's lab in Basel. We've done caloric restriction, 35% reduction food intake, gave rapamycin, and then we did the combination caloric restriction and rapamycin, starting at 18 months, or as a control, we also start with 10. And so I'm going to show you here the data. What you see here is at 10 months at libidom fed, you see the tandem time, the breakage time is pretty low. Now, when we look at the control animals, and so that were 18 months, still kept on the same diet, and we look at 28 months, so it's 10 to 28 months, you see an increase in the time it takes to break these collagens variables, so meaning that's an increase in cross-linking. Now, when we treated the animals with rapamycin, starting at 18 months and look at 28 months, you see a slight reduction at the time it takes them to break the collagen, the tandems. Same thing for caloric restriction, and then in the combination caloric restriction and given rapamycin. And so that really tells you that it is the first model. So again, you start the intervention late, and it's just a slowing of accumulation. And we can plot this, right? So here's the tail tendon breakage of wild-type mice and plaque. This is when you start at 10 months. Here's the intervention at 18 months. And then you can see that, the control animals at 28 months are up here with the time it takes to break. Rapamycin, the combo or caloric restriction is just down here. So basically, unfortunately, it's just a slowing of the accumulation, and we haven't found that there actually any evidence that they repaired or reverse it. And so the hunt is out to find out these mechanisms that can actually repair and reverse these collagen crosslinks. So in summary, I showed you the first part was we can use the matrix type or the ECM composition for drug discovery, and also there's a new important pathway or mechanism that's important for aging longevity. And the second part, I showed you at least a little bit disappointing part that this intervention can only slow the accumulation of cross-linking, and we are looking now to find ways to actually reverse or break these cross-linking parts. And so with that, I'm happy to take questions.
Thank you so much. We have time for one question. Do we have a microphone? Otherwise,
thank you. Hi, very nice talk. I wanted to know whether these macrotypes that may be representing different stages like aging, disease, or health are majorly different as responses or dysregulation. So for example, the differences you see within healthy versus aged states, is this because there's a dysregulation in this organization, or rather is this an appropriate response to the age state, or maybe a combination of both? But do you think one or the other is more important or more prevalent than the other?
It's okay. I understand your question. It's very hard to say because, surprisingly, when you think of one cell, right, but we're multicellular, and it's very well linked. So if you look actually at the EM section, right, it's very, if you ignore the organelles, you see it's very hard to see what is the inside and outside, because all the bundles, the collagens you see, right? You see also the cytoskeleton, and so it's actually linked. So if you pull one side and the other side, so it goes back and forth. And what we think is going on with the mature type is actually that it's just a reflection. It's like mirroring the internal cellular state, right? So meaning that the cell becomes cancerous, it changes the environment, right? It changes all the environment there. So, you know, the collagens have to produce and secrete it. And so it's just a mirroring, and it's all connected or interconnected, so to speak.
We don't have time for more questions, sorry. Maybe you can ask in the break, because we have a break now for 15 minutes. And thanks again, all speakers, for a great session.